Thank you so much for that introduction. Can everyone hear me okay in the back? You okay? It's such a pleasure to be here and to talk with you about my research. I'm going to talk about the human microbiome today, and I know this is a relatively new topic and not everyone might know much about it. So I'm going to begin with an introduction about what it is, why it's important, and why it's important to study its evolution and changing ecology through time. And to explain that, I'm going to delve into some of my work I've been doing on ancient samples, and I'm going to talk both about the gut microbiome and the oral microbiome. So this here looks like a star field, but it's actually a photomicrograph of the human microbiome. And in fact, what it actually is a photograph of human teeth, the surface of a human tooth. And what I've done here is I've just stained the DNA with a blue fluorescent dye so that you can see the hundreds of millions of genomes on the surface of a human tooth, which is about what your teeth look like right now if it's been a few hours since you brushed them this morning. The human body contains an estimated 10 trillion cells and 100 trillion microbial cells. So if you add these two numbers up together, you will find that we are actually 90% bacterial. Now, 100 trillion bacterial cells, that is an enormous number. In fact, it's really hard to conceptualize a number of this size. So let me put it into an analogy. So if you were to take all of the microbes that live just within your own body, and they're on average about 1 micron long, which is about 1/1,000,000th of a meter. And if you were to line them up end to end in a single file line and just stretch them all the way out, they would actually wrap around the Earth twice. That's how many cells that is. So 100 trillion cells is truly an astronomical number. And even this fails to capture the immensity of this number because there are only about 300 billion stars in the Milky Way galaxy, which means that the number of bacteria in and on your bodies outnumbers the number of stars in more than 300 Milky Way galaxies. So at this point, you may be wondering how we appear human at all, and the answer is that while they are numerous, these bacteria are also extremely small. They're about 1,000 times smaller than a human cell. So when you add them all up, they weigh approximately 2% of your body weight, or roughly 1.5 kilograms. This is actually a really important number because that's the same weight as your brain and your liver. And so there are many that have argued that we should start conceptualizing the microbiome as an additional organ system. Now, most of these bacteria live in the gut, in particular in your large intestine. And in feces in particular, they are so concentrated that you actually have around 100 billion viable cells per gram. And if you do the math, that means that with every trip to the toilet, you actually lose about 9 to 13% of your total body cells, which is really amazing. Now, another important body site for the microbiome is the vaginal microbiome. There's about 500 milligrams at this body site. And this is really important because this is how we transfer the microbiome to new generations. So babies are the initial inoculation of an infant with microbes largely occurs at birth. And initially, this transformation, this transfer of vaginal microbiomes colonizes the whole baby. And in fact, no matter where you swab them, if you swab their ears, if you swab their nose, their mouth, their stool, they will all resemble a vaginal microbiome. And this is actually quite important. And the reason for that is the vaginal microbiome is dominated by lactobacilli, which are bacteria that are specialized in digesting lactose, which is the sugar in milk. And so this assists with digestion. Now, of course, today, many babies are not born vaginally. They're born by C-section. And it turns out these babies have a very different microbial structure. And in fact, what they do is they resemble the skin microbiome, which is the first microbiome they come into contact with. And what's interesting here is that for the first 3 years of life, you can actually distinguish whether the mode of birth of a child just by taking microbial samples from different parts of the body. So this is very important inoculation. Another important body site is the oral cavity. There's about 100 milligrams of bacteria in the oral cavity. The oral cavity actually holds a really important role in the history of microbiology. So about 300 years ago, Antony van Leeuwenhoek wrote a letter to the Royal Society of London, and it's the first undisputed description of bacteria. And he actually took that bacteria from his own dental plaque and a few drunks down the street that he got to donate plaque samples to him. And he described very many small living animals which move themselves very extravagantly within his dental plaque. He drew many of these animals, or these animalcules. You can see a couple here, and we can actually recognize them. This is a spirochete here. This is almost certainly Treponema. It's the most abundant genus of spirochete in the mouth. It's this kind of curly shaped bacteria. These here, this figure F, these are very large bacteria. This is almost certainly a member of Leptotrichia or Fusobacterium. These are extremely large microorganisms in the mouth. And so we can actually look at these 300-year-old drawings that he made with a primitive microscope and identify to roughly the genus level what types of bacteria he was seeing. Now, he tried to count them in vain. He said the number of these animals in the scurf of man's teeth is so numerous that I believe they exceed the number of men in a kingdom. Now, if anything, we know this is grossly understated because there are more bacteria on the surface of your teeth than there are humans on Earth, and each and every day we swallow approximately 80 billion bacteria in our saliva. In addition to being extremely numerous, these bacteria have an immense diversity of genes. In fact, on average, they have about 150 times more genes than our human genome. And for this reason, many people are now beginning to reconceptualize what it means to be human, thinking about the role of both the human genome and these microbial genomes together, and conceptualizing the human now as something like a superorganism, like a colony of bees, or more recently, the concept of a holobiont. This is something where you have an ecosystem that is so tightly interconnected that they function as a single organism, like coral. So rather than simply being a leaf on the great tree of life, in some ways we are more like a tree house, a home woven from branches of both permanent and transient microbial inhabitants. And yet we've only very recently come to even notice this large number of underexplored and mostly nameless microorganisms that inhabit the human body. So in fact, it was only in 2001 that molecular biologist Joshua Lederberg coined the term microbiome to signify the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space and have been all but ignored as determinants of health and disease. And this is rather remarkable because we've made immense advancements in understanding the human genome, but we know almost nothing about the evolution and the ecology of the human microbiome. One of the reasons this is remarkable is because they perform basic core human functions. So for example, the microbiome, the gut microbiome in particular, plays a very important role in digestion. We see this very clearly if you look at other animals. So let's take, for example, cattle and horses. They subsist almost entirely on grass. Grass is mostly composed of cellulose, and yet all animals lack the genes necessary to produce cellulases, which break down cellulose. Instead, they rely exclusively on their gut microbes to break down the cellulose for them, and then they absorb the byproducts of that, which are short-chain fatty acids. Perhaps the most extreme example in the animal kingdom is actually koalas. Koalas, as you know, subsist on eucalyptus, which is highly toxic. They have highly specialized gut microbiota that can break down these toxins. It is so important to transfer this gut microbiome very efficiently from generation to generation. The koala joeys actually spend several weeks before they transition from milk to eucalyptus consuming something called pap, which is specialized feces produced by the mother as an inoculant for the developing koala. Now in humans, the gut microbiome also seems to be patterning very strongly with diet and plays a large role in our digestion, although we're still trying to understand exactly how it works. This is from a paper that my lab published last month in Nature Communications. We wanted to understand how subsistence in humans structures the gut microbiome and its functions. And to do this, we looked at three populations. We looked at a hunter-gatherer population in the Amazon of Peru, the Matses. We looked at a highland farming population in Peru, the Tunapuco. They farm things like potatoes. And then we had an urban population in the US. And we compared the structure of the gut microbiome between these three communities. And you can see that they actually are quite distinct, especially this urban industrial population patterns quite far away from the other populations, from these traditional societies which show some overlap, but also occupy different parts of the graph. Now, what's also interesting here, and this has been seen in other studies as well, is that urban industrialized populations have much lower gut diversity than traditional peoples. And this can be replicated in total number of species. We have fewer species in our gut, and those species have lower phylogenetic diversity, meaning they're not as diverse from each other. As I said, this has been seen many times. And this lowering of diversity seems to be related to lowering redundancy. There's a lot of functional redundancy in the gut, but as you lose species, you lose redundancy, and this may be related to the loss of resiliency, because we see in many pathological conditions a further reduction in diversity. So in many obese populations, we see a reduction of genetic diversity or phylogenetic diversity within the gut, and especially in things like IBD, Crohn's disease, and ulcerative colitis, we see dramatic reductions in gut microbiome diversity, and this is associated with high degrees of inflammation and pathology. Another major function of the microbiome is to produce vitamins and to metabolize drugs. So the gut microbiome is the major producer of vitamin K. It's why you don't need to consume it in your diet. Your gut microbiome will make it for you. It also makes a number of B vitamins like folate, which is important for fetal development, for example. It also metabolizes a number of drugs. This is partially responsible for why oral drugs work slightly differently in different people. Probably the most famous example of this is oral contraceptives. Many of you know if oral contraceptives are taken in combination with antibiotics, they're not effective. It's because that bacterial metabolism makes these drugs more absorbable. The third major function of the microbiome is to assist in the maturation of the immune system. This is particularly important in the first 3 years of life. This works in many ways, both directly and indirectly, through, for example, producing short-chain fatty acids, which are used directly as nutrients by the colonocytes, and in myriad other ways. There's many ways in which, if the education of the immune system goes badly or is altered, you can end up with pathological states. For example, periodontal disease is actually a microbial dysbiosis. It's a microbiome disease, essentially, of the oral cavity that leads to runaway inflammation. And finally, the microbiome functions in defense against pathogens. This is seen probably most clearly in the vaginal microbiome, where native populations of lactobacilli maintain a very low pH environment and also a very high amount of hydrogen peroxide. This effectively kills most other types of bacteria and prevents pathogen transmission and reduces STD transmission. But when transferred to other body sites, the microbiome can also cause a great deal of harm. Recent evidence is drawing a very strong link between things like periodontitis and gingivitis and the entry of oral bacteria into the bloodstream with the deposition of plaques in the heart and the initiation of inflammation in cardiovascular disease. So it's clear that the microbiome plays this critical role in human biology, this fundamental pivotal point. And this is why we need to know more about its evolution in ecology if we are to understand what it means to have a normal and healthy microbiome, and also what it means to have a diseased microbiome. And so in my group, one of the ways I'm approaching this is through ancient DNA. Now, most ancient DNA up until this point has focused on two tissues, either bones or teeth. And the main reason for this is that they're very abundant, and that's what you primarily find in the archaeological record. And they contain host DNA. And from that, great successes have been made in reconstructing the genomes of extinct animals, archaic humans, and even plant and animal pathogens. But the problem with this as a substrate is that the skeletal system itself has no microbiome. And in fact, the body decomposes rapidly after death. And so sometimes we do find cases of mummification, but these are rare. They're limited in geographic space and in time. And mostly what we really are left with in the archaeological record is skeletal material. And so how are we going to approach ancient microbiome studies with just a skeleton? Well, it turns out that there are two ways of getting microbiome information from the archaeological record. So one way is sometimes in the case of mummification, especially natural mummification, or in dry caves or in frozen contexts, portions of the gut microbiome or feces can preserve over long periods of time. We call these coprolites, and this is an example of one here. But much more routinely and commonly, the oral microbiome preserves. Your dental plaque mineralizes during your life to form tartar. That's what your dental hygienists clean off of your teeth. And if you don't get it cleaned off, it just persists in this mineralized state, and it turns out that it preserves very well after your death. And so for the past 4 years, I've been focusing on studying these two different ancient microbial substrates to see if we could learn anything about the evolution and changing ecology of the human microbiome. And so next, I'm going to talk about these two in succession. So first coprolites, and then calculus. So Coprolites look like this. In case you don't know what ancient coprolites look like, most people have never seen them. They can be quite different. You can see there's a range of colors. This one's extremely fibrous. You can often see embedded seeds. These are actual intact seeds that are in this one here. But this is what coprolites tend to look like. And these three examples come from salt mines, and this one comes from a dry cave in Mexico. The first question we really want to ask, though, is, well, what is it made of? Is it molecularly preserved, or is it composted? Is it just soil? What does it look like? They look morphologically well preserved. I'm going to focus on these two examples here. One is from a salt mine in Austria, dating to the Iron Age. The other one is from a salt mine in Iran, also dating to the Iron Age. We can use this program. This was developed by Dan Knight's lab. It uses Bayesian statistics to estimate, if you have something like a sink, like a coprolite, you can give it a number of sources and ask, well, what proportion of the coprolite came from one of these potential sources, for example. And these sources are metagenomic data that we've downloaded from public databases. So things like soil that comes from grassland, or soil that comes from taiga or forest, plaque from the mouth, saliva, skin, or gut microbiota. We can say, well, what proportion of the DNA within these substances looks like one of these things? And we can then generate a plot like this and estimate what proportion comes from where. And so these particular samples are extremely well preserved. So this first sample from Austria, we estimate that over 90% of the microbial sequences inside resemble a gut microbiome. This is truly extraordinary. For the Iranian sample, it's slightly less. We have just over 60%. There's a little bit of skin bacteria in there, and that's probably from handling by the archaeologists, but still a really high proportion of this coprolite resembles a gut microbiome. Now, what's really interesting is we can actually break this gut down. As I talked before, traditional peoples in industrialized societies show distinct gut microbiome communities. We can now break that down and show that actually, if you compare it, all of this resembles the traditional gut. We do not see any proportion of this which is mapping onto the industrialized gut. And this suggests that there's been a major change in the gut microbiome that's been extremely recent in human history. Now, if we go a little bit further, we can ask, well, what types of bacteria are here? So it looks like a gut microbiome, but what's actually there? So I call this my A list of fecal celebrities. These are the real workhorses in your gut. These are very common and very important bacteria. These are the bacteria that are in your gut right now digesting last night's dinner. So almost all of you have these in you right now. But there's one that you don't, and this one's been really interesting for us. It's this one here. It's treponema. So when we first got these results, and we started seeing treponema actually a while ago in a Mexican coprolite we were looking at, and we thought it must be a contaminant because treponema had never been reported in the gut microbiome before. But then when we started looking at traditional peoples, it started picking up. So the Matsés have it, the Tunapuco have it, the Hadza of Tanzania have it, rural populations in Venezuela have it. So we started looking at this, and we thought this was really odd. So we started looking really broadly at who has treponema in the gut. And it turns out that all the great apes have treponema in their gut. As I mentioned before, the two hunter-gatherer populations in the world that have been studied both have it, both in Africa, the cradle of humankind, and in South America, the last major human range expansion. We also see it in traditional peoples on both continents, both in Africa and in South America. But the population we do not see it in is we do not see it in Western urbanized populations. We do not see it in US populations, and we do not see it in Europe. It has been systematically lost. So we've been working on this, and we've been trying to figure out, well, how many treponemes are there? Because they're not named, they've never been cultured, we don't know anything about them. And so using the metagenomic data that we had, we assembled partial genomes for them. We think there's at least 5 strains. This just shows you which populations they're in. They're widely dispersed. And now we're working on more deeply sequencing our coprolites to see if we can place them in this tree. They seem to be involved in complex carbohydrate digestion, starch digestion. And we're looking more into what kind of function they may have in the gut and what this loss may mean for our gastrointestinal health. So next I'm going to turn to dental calculus. This is near and dear to my heart. This is what I've been primarily working on for the past 4 years. Again, to orient you to what dental calculus looks like, this is what it looks like. So for example, right here, This is kind of this shelf life. So you can imagine this gap here between the bone and between these the calculus deposits is the gingiva of the teeth. We can take that tooth and we can cross-section it and look at it using scanning electron microscopy. And just to orient you, this is the enamel crown, and this is the tooth root here. This area right here on the outside is cementum. It's microbially damaged. So you can see the kind of darker color here. This is loss of mineralization through microbial action. And this is the pulp chamber here. It once would have been full of blood and nerves and all the things that keep your tooth alive, but after death it also decomposes quite extensively. But this little bit right here on the edge is calculus. We'll zoom in, take a closer look. So again, this is the calculus deposit on the enamel crown here. This is supragingival calculus. Let's take a really close look at it. One thing you might notice right away is that there's a structure to it. It's layered. It almost looks like layers of an onion or rings of a tree. And that's not a false metaphor. It's because it grows in a similar way. So the way that calculus forms is first you get a layer of dental plaque, and then minerals from your saliva will precipitate spontaneously into it, calcifying it completely and solidifying it. And then a new layer of dental plaque forms, and the process repeats. And it repeats over and over and over again, building up these layers through time, so that if this tooth surface is here, this is the oldest layer that would have been formed early in life. Typically, you initiate calculus formation in your early 20s. And then this last layer here is the last layer that formed shortly before death. And so this person died between the age of 45 and 55. So this represents this last layer here. And then you have something that formed in her 20s up here. And so what you have, which is really extraordinary, is a layered, ordered record of her life history through time. This is something we haven't really been able to exploit yet. We don't yet have the ability to micromill out these sections very effectively to look and see if there's distinct changes through time, but I think this is a really exciting future possibility. We can zoom in even more and take a look at exactly the type of bacteria that we have. So these individual little circles that you hear are bacterial cocci. So these are different cells, and then this is a different cell type that you see in this colony over on this side. The white means that it's fully mineralized. So these bacteria have been completely entombed and mineralized within this dental calculus matrix. This is something that makes it kind of extraordinary compared to dentin and bone. Because those are both living tissues, they're porous, they have natural channels and openings, they're vascularized. But dental calculus is not. There's no natural openings to the outside world, and so it's quite protected from environmental bacteria and other environmental compounds that might otherwise assist in the decomposition of calculus. So to put this in perspective, I think that it's important to make a comparison between calculus and dentin and bone, because they're very different. And so one of the things we've had to learn over the years is to treat them quite differently. If you think about dentin and bone, typically, so the cell density within bone is about 1,000 cells per milligram. Usually less, but let's say 1,000. If we do a DNA extraction on that, we will recover approximately 1 nanogram of DNA per milligram of bone that we analyze, or less. And then if we analyze that, the vast majority of that will be soil contamination. It'll be microbes from the soil. And this is some data we've generated showing this pattern. So we took 4 samples from different areas of the world and different conditions, different time periods to show this is a generalizable pattern. So we have a sample that's about 1,000 years old from the Caribbean, a sample that's 4,000 years old from Spain, a sample that's only 100 years old from the Netherlands, and from Nepal. And we cut out a section of the dentin, and then we shotgun sequenced it. So we just sequenced everything that was inside, all the DNA that was present. What we find is that during life, this tissue should be sterile, meaning there should be no bacteria at all. In fact, all of the bacteria, or all of the DNA you recover, should be host. It should be human DNA. But what we instead find is that the amount of human DNA in these dentin samples ranges from about 0.1% to at most 8% in this Nepal sample, which comes from a high altitude, extremely cold and dry environment, so really ideal conditions. If we take that same tooth, though, when we look at calculus, we see quite a different picture. So first of all, it starts off as a completely different substrate. Calculus has an average cell density of 200 million cells per milligram. We've recovered, I have here, I need to update it, at this point it was up to 500 nanograms. We actually broke that record with a 505 nanogram per milligram calculus sample a couple of weeks ago. Truly massive amounts of DNA. To put that in perspective, getting 505 nanograms of DNA per milligram is the same as fresh human liver. You don't get much more cell dense than that, or much more DNA rich than that, rather. But really importantly, too, is we find a very low amount of soil contamination. So these are these same teeth, but here, this time what we've done is we've pulled dental calculus instead, but they're paired teeth, same teeth, and we've just estimated the proportion of oral bacteria. And what we see here is it actually ranges from about 60 to 80%. So we have a very high amount of the bacteria is from the oral microbiome. This is just another way of kind of showing a similar thing in terms of just how much DNA is there. These are some different set of paired teeth where we took a modern sample. So we have about, I think this is about 80 nanograms per milligram. This is fresh calculus from a volunteer, a Swiss dental patient, who kindly gave us some dental calculus to study. And then we also looked at 4 different medieval samples, and what we found was a range of yields for calculus, but ranging from about 5 nanograms per milligram to about 400 here. This is a log scale. But then if you look at dentin from the same teeth, you can see we never are above 1 nanogram per milligram. So radically different amount of biomass in calculus compared to dentin. Now we thought, well, maybe this is just because it's microbial. But we can actually test that. So one of the individuals had really severe dental pathologies. They had a large oral abscess and also a very large carious lesion that clearly was highly infected at the time of death. And so we thought, well, if we sample that carious dentin and we sample that oral abscess, maybe we'll see elevated microbial DNA there. And so this is this tooth, so it has a very large cavity. This is a fenestration here. This is a drainage opening for the pus from the abscess. We sampled the interior here, we sampled the tooth here, and what we found was that actually, no, they show the same amount of DNA as we find in dentin. And this suggests that there's something really special about calculus and the way it forms and the density of the mineral that does protect DNA over long periods of time. But now that we have this, we can start asking really interesting questions like, what is in calculus? What's all there? If we use shotgun sequencing, we can start to explore this. It's about 99.3% bacteria, but we expect that. It's actually because it's made from dental plaque. But there's this 0.7% other that's really quite interesting. About 0.5% of that is eukaryotic. It's a combination of human DNA, but also dietary DNA. We also have a little bit of archaea. At first, this confused us, because why do we have archaea in the mouth? We think of archaea as being associated with deep undersea vents or salt flats. But it turns out there's a couple species of archaea that live in the mouth. They belong to one genus called Methanobrevibacter. The most common one's called Methanobrevibacter oralis. And all of this here that we detected was Methanobrevibacter oralis. And we also have a little bit of virus. Turns out it's all bacteriophage. These are viruses that infect bacteria. But these are quite interesting because, for example, one of the bacteria we found was one called Streptococcus mitis. This is a very common oral commensal, lives in the mouth. And one of the viruses we detected was a virus that infects Streptococcus mitis. So we have not only the bacteria, but a virus that's very specifically preying on that bacteria, so independent confirmation of its presence. We also started exploring proteomics, in particular shotgun proteomics. What happens if you just pull all of the proteins out of calculus and you sequence it and you find out what's there? And it turns out it leads to some really interesting findings, which I'll talk about in a second. But just comparing here, one thing that was really striking, we looked at modern calculus and ancient calculus in parallel, and they show remarkably similar profiles. Most of the proteins are bacterial, but we do see about 10 to 20% are human proteins, and they really are quite interesting, and they're related to immune function. So one of the questions we had when we started this project, we wanted to learn more about pathogens, oral pathogens. We wanted to learn more about periodontal disease. So the oral cavity is very interesting because it's a reservoir for many opportunistic pathogens. So many respiratory pathogens, even neurological pathogens, can inhabit the human mouth. So it's a reservoir for these pathogens. Also, 90% of the world's population will develop periodontal disease during their lifetime. The oral cavity, the oral microbiome, is the only microbiome in the human body that causes disease in a majority of people, suggesting it's in a very dysbiotic state, that it's not in balance. 47% of the US population currently has periodontitis, which is an advanced form of periodontal disease, a severe form of it. And periodontitis today in living individuals is strongly associated with the overgrowth of 3 particular bacterial pathogens. And so we wanted to know, if we looked in the past, if we analyzed ancient DNA from ancient calculus, would we see similar patterns? So we selected individuals that had morphological evidence for periodontitis. So this gap here is here because this bone has actually recessed, which is part of the progression of periodontitis. You can't see it in the photograph very well, but this bone here is very porous, and that's because there was a lot of bleeding at the time of death. And so this person has an active case of periodontal disease, periodontitis, chronic periodontitis specifically, and so we tested this individual and 3 others like her to answer these questions. So we focused on a population from a cemetery in medieval Germany, particularly at the site of Dahlheim. And we pulled out the different sequences. We used many different methods for doing this, using a combination of targeted 16S approaches. We looked at shotgun sequencing. We looked at shotgun proteomics and compared the results. They were largely comparable. They resembled very much what you would see in a modern profile of dental calculus. But really interestingly, if you start to look specifically at what types of pathogens are there, we see some interesting ones. So these 4 here highlighted at the top weren't necessarily causing any disease at the time of this person's death, but they're closely related to strains that do or could. So for example, certain strains of Streptococcus pneumoniae cause pneumonia and also bacterial meningitis. Streptococcus pyogenes, certain strains of it can cause everything from strep throat to scarlet fever to toxic shock syndrome. Haemophilus influenzae, despite its name, does not cause influenza, but it is responsible for a minor respiratory infection. And Neisseria meningitidis causes a subset of bacterial meningitis cases. Now, it doesn't necessarily mean that this individual had a virulent strain, but this gives us a direct way of actually investigating the evolution of these pathogens and closely related organisms through time. We can very reliably recover genomes from these organisms by looking at dental calculus. And of course, we can radiocarbon date the skeletons and know exactly what time they came from. But possibly more interesting are these last ones, Porphyromonas gingivalis, Tannerella forsythia, and Treponema denticola, because these are the 3 pathogens that today are most responsible for periodontal disease. And what we find is we find them at very high abundance in our samples, in our ancient samples. But we wanted to know, however, okay, so we see them in high abundance, but are they higher than you would expect in a healthy person? So to do that, what we did is we looked at the human microbiome healthy cohort. There's 109 published metagenomes for supergingival plaque. We looked at those and we calculated, well, what's the frequency of, for example, Porphyromonas gingivalis in these healthy individuals? The healthy individuals have a frequency of about 0.08%. And we found that in our individuals, they had 1.2 and 7%, much higher than the median. We're really outside of the distribution here. The same for Tannerella forsythia. Our ancient individuals have much higher percentages of these pathogens than you find in healthy individuals, and the same for Treponema denticola. And so what this suggests is that we have tremendous continuity in the microbes that are responsible for periodontal disease over 1,000 years of time. So the same microbes in the past causing periodontal disease are the same microbes causing it today. Now, one of these bacteria, Tannerella forsythia, we actually found so many reads in our data set for this that we were actually able to reconstruct a microbial genome. Tannerella forsythia has 14 known virulence factors. We were able to identify all of them. We also, for one of them, this TFSB, it's a surface protein, and it allows it to escape the immune system. We actually found not only many reads from this gene, but we also found many peptides that were part of the protein that is expressed by this gene. And so we were able to generate reconstructions not only of the gene itself, but of the expressed protein. You might notice that we have a large gap here. At first, this was extremely distressing to me because I couldn't figure out why we couldn't feel this big gap in the reference genome. But it turns out if the Tannerella reference genome here, if you look at what this gap is, it's actually a 48,000 base pair long contiguous region, contains 53 consecutive genes that are part of a conjugative transposon that carries tetracycline resistance genes. And we can see that our sample does not have this transposon. And in fact, we were able to go back and look at the ends of these contiguous here and marry them and close this gap. And we can say definitively that our ancient Tannerella forsythia genome did not have this antibiotic resistance transposon. Now, the other thing that we can learn from these ancient microbiome substrates, which I personally think is really fun, is that in addition to containing the microbiome, they also act as a sink for all sorts of other things, including host biomolecules and dietary biomolecules. So I mentioned before that we found some really interesting human proteins within dental calculus. What we're finding here is primarily proteins related to the innate immune system. We see this both in modern calculus and in ancient calculus. Proteins are really exciting because when you have a genome, the genome is virtually the same in every cell in the human body. But protein expression varies tremendously by cell type. So we can look at this community of proteins that we recover, and we can actually infer what cell types were present. And from this, what we can see is that actually a large amount of this innate immune system proteins that we're finding are quite specific to something called neutrophils. It's one of the innate immune cells that is involved particularly in the first line of attack against bacteria, and is heavily involved in fighting off dental plaque specifically. And so what they do is, this is a picture of one. Here's a plaque biofilm. This is a neutrophil that's combating this plaque biofilm. They contain a number of things called granules. Inside the granule, they make many cell-killing compounds and proteins, and they secrete these proteins against the plaque biofilm, which helps prevent the overgrowth of the plaque. And we found many of these proteins that are produced in these granules within both our modern and our ancient dental calculus samples. We also analyzed the dentin and found it's quite different. Of course, there was blood present originally, so we do find some blood proteins, and we do find a small amount of innate immune system proteins that are really related to the blood that was once present. The vast majority of proteins we find in dentin are actually related to structure and support. It's mainly collagen, things related to the periodontal ligament, and things like that. The calculus also contains a lot of information about food in different ways. So one way is if you just look at calculus under a microscope, you can see bits of food, particles of food. So these are just 4 different things we recovered from some of these samples from Germany. So we have a bit of connective tissue here. This came from some sort of meat product. We can't identify which. This is a phytolith. It's from a plant. It's completely non-specific. We can't say what it is other than it is a plant. But then we have 2 starch granules here. When it's projected, it's a little bit hard to see, but there are some morphological characteristics of this particular one that we can use to assign it to the plant tribe Triticeae, which includes things like wheat and barley. And this one here, this characteristic oval shape with this depression in the middle, is characteristic of the plant family Fabaceae, which includes things like peas. We also identified several dietary sequences within our data set. We have sequences that match sheep, pigs, brassica, which includes things like cabbage and mustard greens, and also bread wheat. And so we came to the exciting conclusion that little change had occurred in German diet over 900 years. But this is actually really important because it made a lot of sense. We knew what people at this time were eating, and the genetic data that we recovered and also the microfossil evidence that we recovered confirmed this. Lately, though, one thing I've become really interested in is milk and dairying. In addition to loving milk and dairying myself, I think it's a really fascinating question. When did dairying arise? How did it spread? Dairy is a really strange thing for humans to be eating. We're the only animals that consume milk after infancy. And we're only able to do that because we have, in one of 2 ways, adapted to milk consumption. We've either altered the milk to remove the lactose, or we have developed genetic adaptations to allow us to digest lactose in adulthood. All mammals can digest lactose in infancy, but we gradually lose this ability as we get older. And so in order to be able to drink milk as an adult, you have to either change the milk itself to remove the lactose, or you have to adapt to be able to produce lactase, the enzyme that breaks it down, throughout your life. Also, milk is really important today at a global scale. So just to put this in perspective, global milk production today is 697 million metric tons. And again, it's hard to imagine what that means. I have no idea in my head what the volume of a metric ton is, so I converted it into a few other units that I thought might help. So that's the same volume as 278,800 Olympic-sized swimming pools, or 217 Walden ponds. That's actually enough milk for every person on Earth to drink a 1/4 liter every single day. But this is amazing because actually most people on Earth are lactose intolerant and can't drink milk at all. And so it makes me really wonder why it is dairy has become so important and why we've focused on it so much. It's become so integral to global diets. And so we started a project focusing on Europe because it's the best studied region for this. And we wanted to ask the question, could we recover anything from calculus that might be indicative of milk consumption? So we focused on a couple of areas. Northern Europe, Central Europe. This is an area that includes parts of Russia and Armenia. And then as our control, we had some 19th century West Africans who we knew drank no milk. So we have 3 different dairying populations and 1 population we know consume no milk. And we looked at individuals across time in different places, ranging from the Bronze Age to the 19th century. This is what it looks like when you begin the process of extracting proteins from calculus. Again, part of the process. And then we have a whole list of things that we found, but among them was one that was really interesting, and it's called beta-lactic globulin. It's a lipokaolin in the calcium protein superfamily, but what's really special about it is that it's extremely specific for milk, and it's only expressed in milk. Also importantly, humans can't make it. We actually have a defective copy of this gene, and we don't express it at all. So the only place that you're going to get it is from animal milk. So these are our results. We tested these calculus samples, and we found that roughly between 15 and 30% of the individuals that we tested positive for beta-lactic globulin in these dairying cultures, where 0 tested positive within the non-dairying culture. But also one thing that's really interesting is because it's protein sequence data, there are sequence variants that allow us to identify what type of milk it is. So when we looked at Bronze Age Italy, we found that it was goat milk that they were consuming. When we looked at Roman era Britain, we found sheep milk and cattle milk. And so we can very specifically identify what types of milk are being consumed in different places. And so our next step, we're very excited to begin, is to start systematically testing Neolithic and Bronze Age sites, both in the Near East and across Europe, and trying to track the spread of dairying across space and time. We're also interested to learn, can we find any dairy-associated bacteria or fungi possibly that might be preserved in calculus? And we also want to better explore the biology, biochemistry, and survival of beta-lactic globulin in different dairy products. And so I think the future of ancient microbiome studies is quite bright. I think it's very useful. We can look at many different questions and begin to reconstruct our ancient microbial past. So thank you very much for your attention. So no, what happens is, so bacteria colonize the surface of the teeth through a complicated process, and they form this thick, active, alive biofilm. And then what's happening is, so your saliva is saturated in mineral, in calcium and phosphate. And the reason for that is so that you can remineralize the enamel in your teeth when you're exposed to things like dietary acids. But what happens is it also mineralizes the dental plaque. And so there seem to be these periodic mineralization events. It's not a continuous process. It tends to be spontaneous and catastrophic, rapid, and it will calcify that layer over maybe a 2-week period. And then a new layer of bacteria, dental plaque, forms, and it may grow for some time. It may be weeks, months, years, and then something happens that triggers a calcification event and it calcifies again. And these build up over time. We're not sure yet how many layers a person makes in their lifetime. We just have a few of these SEM images. I want to do a lot more. But even in modern dentistry, there hasn't really been any research on this. And what's the timing, what's the exact trigger? There's not much known other than the fact that this is the pattern of formation. But once it forms, it doesn't remodel. It gets basically buried. It's almost like if you look at like a Maya temple, where you get a temple and then it gets buried by another one and by another one and another one. It's sort of the same process.
I know that they found with some cultures in the world a lot of like sauerkraut or other things that have been fermented. Mm-hmm. Have you found any evidence of that either in the teeth or in gut microbiomes?
It's a good question. To be honest, we just haven't done a very systematic job of looking at, but it's something I want to start very soon. One of the questions we have though is, are the microbes that are used today in industrial production similar to the microbes that were used in the past? And we don't know the answer to that. So one of the things we're trying to do right now is partner with different farms that use more traditional techniques, or go into partnering with ethnographers that work with remote pastoralists and try to take samples of cultured dairy products or cultured different fermented products to see what the diversity is there, and if it does match the types of microbes that we've essentially domesticated for industrial production.